So we finished up talking about hypertonic, isotonic, and hypotonic solutions. And again, we've got this graphic of red blood cells, of what they'll do in these different types of solutions. If we talk just briefly here, I try to draw up some terrible graphics here. I was trying to move quickly today, and I'm just working from home, remoting into my office computer. So anyway, hopefully this helps, right? So hypertonic, again, that's referring to the solution that you put the cell in. So the extracellular, the space outside the cell, the solution outside the cell. If I take a hypertonic solution, that means that the solution has a higher concentration of the solute than the cell itself. So what's going to happen is the water inside the cell is going to want to get out of the cell into the higher concentration solute outside. So osmosis will occur going out of the cell in a hypertonic. Hyper referring to higher than the cell. The concentration of the solutes is higher in the hypertonic solution than it is inside the cell. The cell will lose water. Okay. Iso, typically in a lot of sciences, when we put the prefix iso on something, that means the same. So isotonic means the solution has the same concentration of the solute as the cell itself. So there's no gradient. So there's no net diffusion is going to occur. So the cell is perfectly fine. Right? As opposed to what we call a hypotonic solution. That means the solution has a lower solute concentration than the cell. So the cell has a higher concentration of the dissolved particles in it. So the water wants to get to the place where there's a higher concentration of solutes. The water wants to get in and dilute those solutes. So the water wants to get into the cells. Just off this diagram here, I don't know if it'll let me move So it's actually showing you that, some of these guys, this guy's bursted here, right? So it's going to, some of those cell membranes can only take on so much water before sometimes they will actually burst. So that's what they're showing you in this diagram here. If I put cells in a hypotonic solution, that means water is going to go into the cells and can cause those cells to rupture. All right. And that's, guys, there's a lot in chapter 5. But these are kind of the highest points that I knew I could crank out and get you guys in a day or so. There's a lot of stuff in there that I don't think is necessarily necessary to test you on in a general biology class. So these are the things that I thought were critical. Go back, review these videos. I'm sorry they're in so many pieces, but hopefully they're helpful between this and the content in the chapter. Hopefully this kind of highlights what I think is the most important parts of the chapter 5. And that's definitely kind of I'm going to be pretty selective what I test you on in chapter 5. First 4 chapters, that's all. It's not that this chapter 5 stuff is not good stuff. There's really good stuff in chapter 5, but I don't think it's quite as critical. I've picked the stuff out and talked to you today about the stuff I think is really critical. So read the chapter, watch these videos, ask me questions. I'll be available in the Zoom channel tonight. Hopefully that helps. All right, guys. Have a good night. Stay safe. Stay healthy. All the things now. All right, guys. Have a good one.